Well, welcome back to Stem Cells from the Sofa, a virtual seminar series hosted by the Stem Cell Network, which is designed to reconnect researchers and bring expert content to your sofa. My name is John Draper. I am the Program Director for Research and Partnerships. And today we are joined by Peter Zandstra on Stem Cells from the Sofa. So some big questions that will be covered here. So cellular therapies are using genetically engineered T cells, including the well-known CAR T cell therapies, have shown tremendous promise as anti-cancer treatments and are attracting considerable scientific and commercial interest. Roadblocks such as treatment costs and scaling production yields of T cells are delaying the widespread availability and adoption of these life-saving therapies. So what new innovative strategies can be employed to tackle these barriers? Well, as I said, today we're joined by Dr. Peter Zandstra, Director of the School of Biomedical Engineering and the Michael Smith Laboratories at the University of British Columbia, to find out the progress his team is making in this area. So as always, during the talk, you can actually ask questions, and that's an important part of Stem Cells from the Sofa. So please do use the Q&A box to enter your questions. The Q&A box will appear. You can ask those questions at any point during Peter's talk, and we'll save those questions for after Peter has finished speaking, and then we will I'll address those questions to Peter. So it's with great pleasure that I introduce Peter. Peter Zandstra is graduated with a Bachelor of Engineering degree from McGill University in the Department of Chemical Engineering, obtaining his PhD degree from the University of British Columbia in the Department of Chemical Engineering and Biotechnology, and continued his training as a postdoctoral fellow in the field of bioengineering at MIT. In 1999, Peter began his faculty appointment at the University of Toronto's Institute of Biomaterials, and in 2016 was appointed university professor, the university's highest academic rank. In 2017, Peter joined the University of British Columbia as the founding director of the School of Biomedical Engineering and as the director of the Michael Smith Laboratories. In these roles, he aims to build programs with deeper interactions between the faculties of applied science and medicine, especially related to innovative research and training programs. Peter is the Canada Research Chair in Stem Cell Bioengineering and is a recipient of a lot of awards and fellowships, including the Premier's Research Excellence Award, the EWRCC Memorial Fellowship, the John Simon Guggenheim Memorial Foundation Fellowship, the University of Toronto's McLaren Award, even the Tiller-McCullough Award as well in 2013. Peter is also a fellow of the American Institute for Medical and Biological Engineering and the American Association for the Advancement of Science. So he's extremely well connected, well appreciated for the work that he's been doing, and Peter's research indeed focuses on understanding how complex communication networks between stem cells and their progeny influence self-renewal and differentiation, and how this information can be applied to the design of novel culture technologies capable of controlling cell fate. What actually wasn't in Peter's bio is the extensive leadership Peter has taken within the Canadian regenerative medicine and stem cell research field, and also the commercialization endeavors and the companies that he has been involved with. And just to name a few, Medicine by Design, CCRM, Excelthera, Notch Therapeutics. Peter had a hand in all of these and others as well that I'm not going to mention. So Peter, thank you for joining us today. Great to see you. I hope you're doing really well. Please take it away and start your talk.
Great. Thank you very much, John. It's a real pleasure to be here. I always am excited about the opportunity to talk to and work with the Stem Cell Network. Let me just make sure does that look okay on your side?
Yeah.
And it's you know, I think this format is really exciting because it gives us an opportunity to have a bit of a more hopefully casual discussion on where we are and where the field is going. So looking forward to the discussion part of the session. So just a little bit of disclosure, as John mentioned, I'm involved in a few companies, and some of the data that I'll show has been licensed to these companies, and therefore it's important to recognize that I have that conflict of interest and bias when I present this work. My lab has been interested for since the beginning, really, in using engineering approaches to control cell behavior. Our premise is basically that one can engineer the environment around the cell, or the so-called niche, to control cell fate decisions, and that this will be an enabling element for technologies which are based on stem cells, whether they be stem cells themselves or their differentiated tissues or cells, or even using those cells to discover new drugs, which then can manipulate cells of self inside our bodies. We really have two approaches to manipulating the stem cell fate. One that is the predominant one I'll talk about today, although I'll allude to some other work we're doing, is engineering the niche. And that gives us the opportunity to ask, what is it about the environment around the cell that we can control and mimic, and allow that to guide stem cell responses. We're particularly interested in applying this approach in the context of blood cells as therapeutics for a variety of different diseases. Blood cells, I think, are particularly a great opportunity for a number of reasons. First and foremost, they allow us to move fairly quickly from experimentation and development to therapy. And that's because the path for how blood stem cell transplants, and indeed now more mature functional cells derived from stem cells to the clinic, is well established for unmanipulated cells and now some manipulated cells. And that means that new innovations that we can put onto this system can really move quickly. The other part is that I think the blood forming system represents a paradigm where we can really use that understanding to think of blood cells as delivery agents, as immune effectors, and as different types of vehicles for secondary therapeutics, which might be either directly or indirectly related to blood diseases. And finally, as you'll see, blood cells are an interesting model system to look at in terms of different developmental sources, whether they be the blood cells which are derived from bone marrow or peripheral blood in the adult, from the cord blood in the neonate, or even from early developmental stages such as we're deriving from pluripotent stem cells. And each of these different sources have different strengths and weaknesses and different advantages in terms of how quickly we can move and what we can learn. So given that, why has it been so difficult to grow blood stem cells and to control the generation of functional blood cells from these stem cells? And we very early on started to look at the types of cells which are formed when one puts cells into culture, and we recognized that, as many others have, that when you start with a stem cell, whether or not you have perfect culture conditions, you quickly produce other cell types. And these other cell types might be cell types you're interested in, or they might be cell types that interfere with the further growth and differentiation of those blood cells, creating feedback loops which interfere with our ability in vitro to control the environment and generate the products we want. We've applied this knowledge to a number of different areas. This slide just reviews some of the published work. And I wanted to include this slide because I think this is a really exciting demonstration of the power of the Canadian Stem Cell Network to move fundamental science into clinical therapy in a rapid way. The early work by Liz Kassar to show us that feedback control would be important for helping us enhance the growth of bioreactors was in 2013. Eman Ferez, working with Guy Sauvageau and Anne Marigny, then developed a molecule which changes the composition and rate at which these cells differentiate, further reducing feedback in these cultures. And then working with Sandra Cohen and others at the Hôpital Maisonneuve Rosemont in Montreal, we've now shown that the cells grown using these molecules and these culture technologies can have a great impact on diseases such as AML. And again, this work was strongly supported by the Stem Cell Network. So today I wanted to focus on another story, which is now that we have better and better ability to generate or characterize stem cells derived from cord blood, what about the cell types which those cells can produce, and what cell types might we start working on to generate from cord blood and other stem cells? And clearly an exciting potential area is the lymphoid branch of hematopoiesis, where T cells are made. T cells have direct interest in transplantation as sources of cells that might overcome lymphopenia. We know, and we learned from our work with cell transplantation of stem cells, that T cells are very involved in modulating graft versus host disease and graft versus leukemia targeting. But we also know that mature cells are being isolated from patients every day and transformed into use as immunotherapeutics. And it got us thinking maybe we could use stem cells as sources for T cells and really start to enable a broader and more strategic use of these cells to overcome some of the challenges that currently are facing the immunotherapeutic area. So just to review, as John mentioned, T cells are a very exciting therapeutic. Typically, they're isolated from patients through leukapheresis. They're transduced in vitro with targeting moieties, chimeric antigen receptors, for example, and then expanded with beads and other technologies outside the body before they're infused back, where the targeting moiety then guides the T cell to act against a specific cancer, usually a blood cancer, in most cases a B-cell lymphoma. So this has been a great technology, but in order to really broaden its use, adopt it for new types of cancers, and make it accessible to Canadians and people across the world, there's a number of challenges we need to overcome. First of all, because it's a patient-derived product, it's highly variable. Second of all, because each product needs to be transduced with virus in vitro, it adds a lot of cost and variability. And finally, depending on the patient and depending upon the in vitro culture technologies that are used to expand it, there's a lot of heterogeneity. And I think this is one of the big challenges that the field faces right now. So with that in mind, a number of years ago, we started collaborating with really the world leader in developmental immunology, Juan Carlos Zuniga-Fluker from Sunnybrook and the University of Toronto. And we started thinking, given what's known about mimicking T-cell development in vitro, can we start to enable new technologies that allow us to do this in a clinically relevant, controlled, and enhanced manner? Now, Juan Carlos' lab had developed a new culture technology called the OP9 culture system for a while. This is an engineered feeder layer that expresses a notch ligand on the surface of the cells. And this is an important and critical signal that occurs in the thymus, here shown in this kind of pinky peachy type box, that is important to guide T-cell development from early derivatives of blood progenitors as they enter the thymus and then migrate through different locations along that way. And we wanted to explore whether or not we could start to define a minimal set of conditions which would allow T-cell development in vitro from these human cord blood cells. And this is work that was done by Shreya Shukla when she was in the lab, and assisted through collaborations in the lab with John Edgar and Matthew Langley. And what Shreya found was that if she immobilized DL4 alone onto surfaces, she was able to get some development of early progenitor T cells, but really this wasn't very robust and the viability wasn't very good. However, when she was added different extracellular matrix proteins, she started to have positive effects. And to make a long story short, and the details are in this paper here, she eventually identified VCAM, or vascular adhesion, VCAM1 as a molecule which synergized with DLL4 to significantly enhance protein expansion. And you can see that here on this plot specifically, where you see significant increases, about 10% at this stage, in progenitor T cells relative to when you have DLL4 alone. Shreya also identified what the mechanism of this was. We showed that what was happening was that the presence of VCAM on the surface increased T cell migration. That migration resulted in increased pulling against the Notch ligand, which was also immobilized. This increased pulling increased Notch target gene activation, and that subsequently enhanced T cell differentiation. We know that the progenitor T cells produced through this in vitro technology were functional when transplanted in vivo in neonatal mice, and this is work that was really done through a collaboration with JC's lab by Jasti Singh. And what Jasti helped us show was that when you transplanted these progenitor T cells, either when they were made in control conditions on OP9 or when they were made in these conditions with DLL4 and VCAM, they were able to differentiate in vitro, they were able to respond and secrete cytokines, and they looked and acted like mature T cells through this process of in vivo development. This is where we start to think, well, now that we have a minimal set of conditions where we can have in vitro differentiation of T cells, what can we do to really enhance this process and start to mimic some of the other signals which occur during thymic development? And John Edgar's work now has shown that a number of new molecules synergistically enhance this T cell lineage differentiation, both in terms of the yield of cells as well as their subsequent maturation. And you can see some of that data on this slide here, where our original condition without IL-3 and TNF is shown here, and both in terms of total cell expansion, in terms of progenitor T cell expansion, you see a significant enhancement both in the purity and the yield of this cell upon addition of IL-3 and TNF. You know, we think there's some really interesting mechanisms behind this that I'm happy to talk about in the question period, but one of the things that I think is particularly compelling is the role that these molecules have in reducing the dependence of T cell development on the amount of Notch signaling. And you can see that here, where under control conditions, you see that DL4 alone, you get very little protease development, and you get, as we showed earlier with DL4 and VCAM, you get nice progenitor T cell development. With IL-3 and TNF, you even get T cell development in the absence of VCAM, and then a further enhancement. And then if you look at the amount of DL4 needed for this differentiation in a dose-dependent manner, we can significantly reduce this. Now, why is this important? This is really important when we think about the types of manufacturing we want to do, because figuring out how to present DL4 on surfaces in a scalable manufacturing aspect is a significant challenge to scaled clinical protocols, and this really helps. This will really help us along the way. John has also shown that the progenitor T cells made in this system can be transduced and matured into functional TCR expressing CD3, CD8 positive cells. And this is shown on this plot here, and that shows that these in vitro cells, although they don't go through all the thymic selection steps, we're able to start to recapitulate and reproduce some of those steps by inducing new synthetic molecules to the surface of these cells. And this, you can see this comparison here between the non-transduced cells and the transduced cells in terms of the more mature cells which they produce. So this is where we were a few years ago, and some of John's data more recently. And then we started to think that indeed making these cells from cord blood represents a great opportunity. But if we really want to start to impose controlled engineering of cell fate upon these, wouldn't it be great to start with a cell type which is more like a cell line, and see if we could generate blood cells and eventually T cells from these so-called pluripotent stem cells that are derived from earlier on in development. And so if we go back to the original slide that I showed you, what we've added is a whole bunch of new steps of complexity. Before, we were looking at blood stem cells, generating mature cells, and how to grow these blood stem cells. Now we need to back up several steps right to pluripotent stem cells, and they go through a number of different stages of development, perhaps generating mesoderm, hemogenic endothelial cells, blood progenitor cells of the right type that can eventually produce T cells or other cell types according to those different pathways. And we've been working on this area for a number of years to try to understand what the signals are that one would need to mimic, or the niche of early blood cell emergence. I always show this picture from the DERIVED lab, which is really the emergence of early blood cells in an avian system. But just to show you how dramatic that emergence of blood progenitors is in this out of hemogenic endothelium early during development. Of course, we and others have investigated for a number of years what the signals are, autocrine, paracrine, inflammatory molecules, and other types of molecules which might enhance the right type of hemogenic endothelium to produce the right type of blood progenitors that are capable of lymphoid development. And some work that I'll allude to here was some work by Nafis Rahman in 2017, where Nafis showed that by controlling the types of aggregates which were made using the Agrowell technology, which Mark Ungren developed in our lab, and then by studying different media components upon using plated and patterned hemogenic endothelium on surfaces, he could start to identify cytokines and growth factors which would enhance this endothelial to hematopoietic transition period. This allowed us to start to combine different technologies along the way, where we might start with iPS cells either in their normal phase or genetically tailored, use technologies which have emerged out of the lab or other labs, such as the Agrowell differentiation systems, our artificial thymic niche based on DL4 and VCAM, to guide the different stepwise development. And this is really work that's now led in the lab by Yale Michaels, and has been a really exciting development for us over the last few years. And one of the things that Yale has shown is that when you start to apply these technologies and combine our knowledge from growing enhanced numbers of blood T cell progenitors from cord blood, for example, using John's IL-3 and TNF media, with our ability to induce blood cells from pluripotent stem cells, we can also significantly enhance this process. And this is just some data showing CD5, CD7 progenitor T cells generated in the media that has, for example, TNF alpha and IL-3, showing that even at low cell densities, we can generate nice amounts of progenitor T cells, and that over time, this further gets enhanced and accelerated, so that these cells are generated with really good yields as well, compared to technologies which don't use some of these media type systems. Yale has also shown that these cells can be further differentiated into, shown here, CD4 immature single positive cells, just focusing on one plot on this fairly complicated slide, showing that under normal conditions we can probably get a several-fold increase in the number of ISP positive cells generated at low cell densities, which allow much higher yields and easier manufacturing design than the established technology here shown as media B along the way. And this has opened up all kinds of things for us. It has opened up the idea that maybe we can take some of these signals that we're applying to cells in vitro and bring them in vivo, and Cell is working on this in the lab, generating new hydrogels to allow this. It's allowing us to think about how we've shown now that we can generate cytotoxic T cells of different types, but how can we guide the process of this progenitor T cell differentiation to generate different types of T cells along those pathways? And of course, it's enabling us to study thymopoiesis and human T cell development in a way that's not been able to before, because we really have scalable access to all these technologies out of pluripotent stem cells, and those cells themselves are suitable for genetic engineering and editing. So where is this all going? This is really a platform that we're really excited about. We can generate, as we know from other work in our lab, clonal iPS cells with specific properties. We can then impose these properties upon cells, whether it's targeting moieties, different types of ways of controlling bifurcations and cell fate decisions, or different advantages based on the types of cells we select or how they're transplanted, and differentiate these into functional cells that hopefully will be enabling for a variety of clinical therapies, both those that are currently being used as well as new ones that will only be accessible with this additional functionality that we're starting to impose on these systems. And this is really pointing towards the other side of the work in the lab, which is what is the functionality that one might want to put in these progenitor cells and in these pluripotent stem cells as one guides these towards the programmable and predictable behavior which one wants. And just some examples of that shown here from some work from Jonathan Lifshitz, which he published in 2017, where we're matching both manufacturing challenges as well as potency challenges to cell products through these differentiation paradigms represents a great opportunity. One can think of enhanced potency if you want to think of ways of expanding subpopulations of cells or maturing them. One can think of metrics which allow us to rapidly and cheaply indicate the quality of the in vitro generated cell population. And finally, one can think of ways of engineering these cells to reduce the costs of manufacturing, whether that's getting them to respond to different engineered ligands in these bioreactors, or even get them to produce some of these differentiation moieties themselves to enhance that through feed-forward loops, which might be very exciting. And this is an area, actually, I should mention that this interface between bioprocess engineering and synthetic biology, I think, represents a great opportunity, and certainly one where we're looking for students specifically that have a process engineering background that are interested in working in this area. So please reach out to us if you're interested in that. That's really what I wanted to talk about today. I think we're more or less on time. I did want to, first of all, I tried to acknowledge everybody who did the work along the way, but in fact, we're a big cohesive lab, and almost everything we do is through internal interactions and support, and I'm very thankful for the group we have at UBC and all the great work they're doing. This is a picture from one of our retreats recently, where we were up at a lake in the forest during a rare rainy day. I'm also thankful for the support which we've received from the Stem Cell Network, CHR, Leukemia, Lymphoma, NSERC, and CFI for helping with our work. And I'll finish with this slide, which is particularly poignant today because today is the PhD defense for Jennifer Ma, and these are some pictures from different covers from stories from our lab that she has made, and the one in the middle particularly talks about this story about DL4 and VCAM and the way it signals through cells to enhance T-cell development. So happy to take questions.
Fantastic. Thank you so much, Peter. Great talk. So yeah, please do ask put questions in through the Q&A box, and I can start you off with one. And this is something I certainly was familiar when I was doing stem cell research. The maturation process from cells that are derived from pluripotent stem cells, that can be tricky, especially within the blood system. This has bedeviled the production of, for instance, red blood cells. Does the same issue factor into making functional T cells?
Yeah, I mean, certainly the ability to mature the cells is an important thing that we're working on. What I would say is because these cells are going to be engineered anyways for their clinical efficacy, there are really interesting synergies between the molecules which we're putting on the surface of the cells and their increased maturation and function. For example, we know that signaling through T-cell receptor is an important maturation signal for T cells, and that many of the chimeric antigen receptors which are put in these cells actually have components of signaling pathways which mimic T-cell receptor maturation. So we think there's great opportunities there for taking advantage of both the maturation signals, which are necessary, but also the therapeutic moieties, which are important for the ultimate use of these cells.
That's fantastic. And it shows this real promise then, right? It's quite hopeful.
Yeah.
So what about autologous therapies versus allogeneic in this context? Do you envisage using these to bank T cells, or are you imagining this as a personalized medicine approach?
No, we really are looking at an autologous approach where we can bank cells, an allogeneic approach where we can bank cells and use them on demand. There's a number of reasons why this should work really well based on T cells. First of all, there's already clinical evidence that can work from companies such as Allogene, but we also know that we can both the duration of even under certain circumstances, the duration in which the cells need to be around is quite short for them to have good clinical efficacy. So even without further engineering the cells to allow and control their immune activation, we think that will be good use as an allo product.
Very cool. Okay. What about the scaling challenges? I mean, this seems to be something that is a huge piece of work and endeavor to actually get together to get these to the numbers that are going to be required for transplantation. What are the major hills that are still to climb there?
Yeah, I mean, that's as you know, that's my bread and butter, and that's something that we've been thinking about since the very beginning. I'm really excited about the ability in which we can interface the technologies I talked about today with scalable manufacturing processes. And we know we have good sense of how many cells will be needed because we have control. In the clinic are autologous derived cells right now, so we know what our targets are, and we know how to characterize these cells in terms of what's been used for the autologous targets. So I'm confident even today, without significant further innovation that we're actually working on, that we're going to be able to hit the types of numbers that are necessary to put this into people.
Okay. So a question, does the T-cell maturation process need to be confined to a surface? Would that inhibit scalability?
Yeah, the notch ligand is a ligand that requires a pulling type activation against its receptor to start that signaling pathway. And that typically requires adhesion to a surface or some sort of a structure in order to do that. And I think that's where a lot of really interesting innovation is happening. How can we design surfaces or moieties which allow the cell to pull against the Notch receptor while also doing it in a manner that allows for scalable expansion? And you know, a number of people are working on that, including folks in JC's lab and in other places. So it's an exciting area, and I think you know, there'll be great results coming out on this soon, I'm sure.
Fantastic. So keep the questions coming in if you've got any more. So there is a question about transplantation costs. You mentioned that this is one of the aims is to reduce transplantation costs. I'm currently ignorant about what the current technology in terms of CAR T cells therapies, what they actually cost, but how much do you think you can reduce it, and how will that benefit consumers or patients?
Yeah, I mean, it's always hard to put a number on overall costs without understanding the full kind of chain logistics that are important here. What I can tell you is that the current costs are really quite significant. You know, Chimera, Yescarta are typically in the $400,000 US range, and that doesn't include all the hospitalization costs and other ancillary costs which sometimes come into these therapeutics. We think that but you can imagine that those costs are significantly driven higher because they are patient-specific therapeutics. So everything has to be done, and all QC has to be done on each patient. Whereas if you can have an allogeneic a product where you have characterization and manufacturing, and you can have off-the-shelf products, we believe that will significantly reduce these costs and increase accessibility, while also increasing the quality of the product, because we can do so much more characterization and engineering around its properties.
I see. So there's one more question here. Are there any concerns on the immunogenicity of patient-derived CAR T cells?
So I'm not sure I completely understand the question, but I'll try to talk through it. So certainly, you know, with patient-derived CAR T cells, typically these cells will be taken from a single individual and given back to that individual, and they'll have enhanced immune function because they'll be engineered to express a receptor, a chimeric antigen receptor, where that receptor would recognize unique antigens on the surface of cancer cells, which hopefully will have a positive outcome. So we are purposely enhancing, I guess, the immunogenicity of those cells, but not in a way that would enhance their rejection, but really their killing ability for specific moieties within the patient.
Okay, I think, oh, one more. Why use embryonic stem cells as the source as the pluripotent source rather than iPSCs?
No, iPSCs are a good source of cells. There's no, I mean, in fact, that's the source we're predominantly using. So.
Fantastic. Okay, well, with that, Peter, then I will say thank you very much for joining us and giving a great overview of the work that you're doing. Wish you well, and hopefully this work can get to the clinic as soon as possible.
Thank you very much.
Oh, sorry, Peter, sorry.
No, thank you. It's a pleasure.
In the meantime, before everyone leaves, I just want to talk about the next event from Stem Cells on the Sofa. Wednesday, September the 9th, at 1 p.m. Eastern Time. We're joined by Frida Miller, who's a professor and senior scientist at the Hospital for Sick Children and the University of Toronto, who is internationally recognized expert in the study of neural and dermal stem cells, as well as the nervous system development. So Frida will be discussing recent work identifying the mechanisms that regulate establishment of postnatal neural stem cell pools and then that then allow the genesis of specific types of mammalian neurons. The possibility that these endogenous precursors might be recruited to promote human pediatric brain repair will also be discussed. And so make sure you register for that. And in the meantime, you can connect with us via the website, stemcellnetwork.ca, or Twitter, which is @stemcellnetwork. And it's great to, oh, and the video of this will be posted on our YouTube channel as well. So Peter, thank you very much for joining us. And until next time, thank you and bye-bye.